Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the Deep Dive. Today we're tackling one of the biggest health trends out there right now, something you've almost certainly heard about: time-restricted eating.
Right. It's a cornerstone of intermittent fasting. And the appeal is huge, isn't it? The idea is just so simple.
Exactly. You don't need to count calories. You don't need to change what you eat. Just limit when you eat. Squeeze it all into a shorter window each day.
And the promise is that this simple time constraint, maybe eating in an 8-hour window, like say 10 a.m. to 6 p.m., will somehow flip a switch in your body.
Yeah, proponents claim it improves your metabolism, makes you more sensitive to insulin, helps you lose weight, all because of the timing.
It sounds almost like a magic bullet, which is why it became so popular so fast.
That simplicity is definitely powerful. But the reason we're diving in today is a brand new study, a really rigorous, very highly controlled study that, well, it throws a pretty big scientific wrench into that simple narrative.
We're talking about the ChronoFast trial. This was a collaboration between the German Institute of Human Nutrition, DIfE, and the Charité University Hospital in Berlin.
And this trial was specifically designed to answer a crucial question that previous research kind of glossed over.
Exactly. You see, lots of earlier studies showed benefits with time-restricted eating, but they couldn't clearly say, were those benefits because of the timing itself? Or was it just because people sort of naturally ate less when they had fewer hours to do it?
That's the key confusion, isn't it? If you suddenly stop eating at 6 p.m. instead of 10 p.m., you're probably cutting out snacks, maybe dessert.
Yeah.
You're likely cutting calories without even trying.
Right. And that accidental calorie cutting, what the researchers call spontaneous calorie restriction, we know that leads to weight loss and metabolic improvement.
Yeah.
So was it the clock or the calories?
And this is where ChronoFast comes in. The researchers set up this incredibly meticulous experiment to isolate the effect of when you eat from how much you eat. They made sure calorie intake stayed the same.
And when they did that, when they controlled for the calories and only changed the timing of the eating window.
Well, the results were, let's call it a metabolic reality check.
Pretty much. The restricted eating schedule on its own, even when they tested an early window, which is often hyped as better, it just didn't produce those measurable improvements in key health markers like insulin sensitivity or cholesterol.
Okay, so this is a really significant finding. Our mission today is to unpack this ChronoFast trial. We need to understand just how controlled it was, why that control matters, and what this means for, how we should think about time-restricted eating. Is it really a metabolic key, or is it more like a really effective way to structure your day to maybe eat less overall?
Before we get into the nitty-gritty of the results, let's just clarify the terms. Time-restricted eating, or TRE, as used in this study, means eating within a window of 10 hours or less each day.
Which means you're fasting for at least 14 hours straight. ChronoFast actually used an even stricter 8-hour eating window.
Right. And that fasting period, that 14-plus hours, that's where a lot of the proposed benefits are supposed to come from. Things like improved cellular cleanup processes,
And we should acknowledge the initial excitement wasn't baseless. There were earlier studies suggesting real benefits: better insulin sensitivity, lower glucose, triglycerides, cholesterol improvements, even some weight and fat loss.
Absolutely. But the big caveat, the thing ChronoFast was designed to fix, was the inconsistency in those results and, crucially, the lack of isocaloric control. Isocaloric just means keeping the calorie intake the same.
Without that control, you always have that confounding variable. Did the timing help, or was it just the fact that people ate less food?
It's basic human behavior, really. Shorten the eating window, especially cutting off evening eating, and most people will spontaneously drop some calories. That leads to weight loss, and weight loss improves metabolism. It's a known pathway.
So ChronoFast, led by Professor Olga Remek and her team, set out to untangle this. They wanted to achieve that gold standard in nutrition research. A setting where the only thing changing was the timing of the meals, not the total energy consumed.
They wanted to finally separate the clock from the calories.
Okay, let's dig into section 1, the ChronoFast study design. You said it was incredibly rigorous. What made it so?
Well, the core goal was clear. See if an 8-hour eating window improves things like insulin sensitivity when you take calorie reduction out of the picture.
And the structure they used, a 10-week randomized crossover trial, that's a really strong design, isn't it? Can you explain for our listeners why that crossover approach is so valuable here?
It's super powerful, especially in nutrition. In a typical study, you might put group A on one diet and group B on another, but people are different. Their genetics, their baseline health, these things can affect the results.
So you get a lot of noise, basically.
Yeah.
Individual differences obscuring the real effect.
Exactly, but in a crossover trial, every single participant does both interventions. So in this case, everyone tried the early eating schedule and everyone tried the late eating schedule.
Ah, so each person acts as their own comparison point.
Precisely. You compare how that specific person responded to early eating versus late eating and versus their own baseline. It cancels out a huge amount of that individual variability, making the findings much more reliable.
Okay, so the trial structure involved what, a baseline period first?
Yeah. They started with a 2 to 4 week baseline phase. This was just to see how participants normally ate and what their baseline metabolic health looked like.
Then came the interventions.
Then came 2 separate 2-week intervention periods, one where they followed the early eating schedule, one where they followed the late one.
And crucially, there was a break in between.
Yes, a 2-week washout period between the 2 eating patterns. This is important to make sure the effects of the first pattern didn't carry over and influence the results of the second one. Let the body reset to baseline.
Got it. And who are the participants in this trial?
They recruited 31 women who were classified as having overweight or obesity. Their average BMI was around 30.5, and the median age was 62. So mostly postmenopausal women.
That's a very specific group. Important for studying metabolic health, as you said, because this group often faces challenges with insulin resistance.
Right. But it also means, as you hinted, we need to be careful about applying these exact findings to, say, young, lean men or other populations. The study's strength here was its homogeneity, keeping the groups similar for better control.
Okay, and the 2 eating schedules they tested, both 8-hour windows.
Correct. Both strict 8-hour windows. The first was early time-restricted eating, or ETRE. That meant eating only between 8 in the morning and 4 in the afternoon.
That's one often claimed to be metabolically superior, right?
Right.
Aligning with supposed natural rhythms.
That's the theory, yes. The second was late time-restricted eating, LTRE, eating between 1:00 p.m. and 9:00 p.m.
So, same duration window, just shifted later in the day. And how well did people stick to these schedules? 8 hours is pretty tight.
Amazingly well, actually. The researchers reported over 96% adherence to the timing schedules, which they tracked using detailed food records. That level of compliance in a study where people are living their normal lives is, well, it's remarkable.
Okay, adherence was high, but the real challenge must have been the isocaloric part, keeping the calories the same. How did they manage that when people suddenly had way less time to eat?
It was a multi-pronged strategy. First, the instruction wasn't just eat in this window. It was eat your usual amount of food, but fit it into this window.
So actively encouraging them not to cut back.
Yes. And they used detailed food records monitored with a digital app to track intake closely. Plus, participants received specific dietary counseling focused on how to condense their normal day's worth of food into those 8 hours without changing the total energy.
That sounds like it would require some planning from the participants.
Definitely. They also monitored the macronutrient the balance of fat, carbs, and protein to make sure the quality of the diet wasn't shifting either, because that could also affect metabolism independently.
And they controlled for other factors too, like
Well-
exercise, because if one group started moving more, that would skew things.
Absolutely. They used motion sensors, actigraphy to track physical activity levels and ensure they stayed consistent across all phases of the trial. Baseline, early eating, late eating. They were really trying to lock everything down so that the only significant variable changing was the clock time of eating.
And the measurements they took were pretty comprehensive, going beyond just asking people how they felt.
Oh yeah, very thorough. They used continuous glucose monitoring, CGM, which gives you a 24-hour picture of blood sugar levels in real life, not just a single snapshot in the lab.
That's becoming more common, provides much richer data.
It does. They also did the standard oral glucose tolerance test, or OGTT, which is the classic way to assess how your body handles a sugar load, and of course, detailed blood tests.
Looking at what's specifically in the blood.
Insulin levels, a full lipid panel cholesterol, triglycerides and markers of inflammation like high-sensitivity C-reactive protein and interleukin-6, all the key players in cardiometabolic health.
Now, you mentioned something really cutting edge. They actually measured the body's internal clock.
Yes, this is what really elevates the study, methodologically. They used something called the body time assay. This isn't just about sleep diaries, it's a molecular test. They looked at biomarkers, specific gene activity patterns in blood cells, monocytes, to get an objective reading of the individual's internal circadian phase.
Wow. So they could literally see if shifting meal times from, say, ending at 4 p.m. versus 9 p.m. actually moved the timing of the body's internal biological clock.
Exactly. It allows them to directly measure the chronobiological impact of the eating schedule. The level of detail and control here is really top-notch nutritional science.
Okay, so that sets the stage. Incredibly controlled study design. Now, section 2, the core finding. What happened to metabolic health when they isolated timing like this?
Well, this is where the hype kind of meets reality. The main finding, the primary outcome they were looking for, showed basically nothing. No benefit from the timing alone once calories were controlled.
And that primary outcome was insulin sensitivity, right? Measured using the Matsuda index. Remind us why that's such a key marker.
The Matsuda index is calculated from glucose and insulin levels during that OGTT. Essentially, it reflects how efficiently your body uses insulin to manage blood sugar. A higher score means better sensitivity. Your body responds well to insulin.
And low sensitivity, or insulin resistance, that's the big problem leading towards type 2 diabetes and metabolic syndrome.
Precisely. So if time-restricted eating really has this powerful metabolic effect through timing, you'd expect the Matsuda index to improve significantly, especially maybe with that early eating window.
But it didn't.
No. The results showed no difference in insulin sensitivity between the early eating pattern and the late eating pattern. And maybe even more importantly, neither pattern showed any clinically meaningful improvement compared to the participants' baseline habits.
So squeezing their eating into 8 hours, whether early or late, didn't make their bodies better at handling sugar as long as the calories stayed the same.
Correct. And they double-checked this using another established measure, the Oral Glucose Insulin Sensitivity Index, or OGIS. That analysis confirmed the same thing, no change.
That's a pretty direct hit to the idea that timing itself is the magic ingredient for insulin sensitivity in TRE.
It really is. And this lack of effect wasn't just isolated to insulin sensitivity, it held true across pretty much all the other metabolic markers they measured.
Okay, what about blood sugar control over the whole day? Did the continuous glucose monitoring show anything?
The average 24-hour glucose levels were not meaningfully affected by either the early or the late eating schedule. Daily glucose control seemed largely indifferent to the timing, provided the energy intake was constant.
What about heart health markers? Cholesterol, blood pressure, common targets for TRE claims.
Same story there, unfortunately for the hype. Total cholesterol, LDL cholesterol, the bad kind, triglycerides, systolic and diastolic blood pressure, none of them showed any significant changes between the eating patterns or compared to baseline.
Even inflammation. Those markers like C-reactive protein.
Unaffected. The inflammatory markers also didn't budge.
Wow. The consistency across all these markers, glucose, lipid, blood pressure, inflammation, really paints a clear picture, doesn't it? If calories are matched, the timing alone seems to have very little impact on these core metabolic health indicators.
It's a strong message from this data. Now, there were a couple of minor changes they noted, but they don't really alter the main conclusion.
Oh, there were those?
HDL cholesterol, the good cholesterol, actually went down slightly in both the early and the late groups.
A decrease in good cholesterol isn't exactly a benefit.
No, it isn't. And also, a liver enzyme called gamma-glutamyl-transferase, GGT, it also decreased slightly in both groups. Sometimes lower GGT is seen as positive, maybe related to liver health or oxidative stress.
But these were minor changes.
Very minor, and the researchers stressed that the overall cardiometabolic health picture remained essentially unchanged in any clinically relevant way.
Okay, this brings us back to that crucial factor, the calories. They aimed for perfect isocaloric control. But you mentioned earlier it's almost impossible to achieve perfectly in real life. Did a small calorie difference creep in?
Yes, and ironically, this slight imperfection actually reinforces the main point about why previous studies might have seen benefits. Despite the intense effort, a small energy deficit did occur.
Was it different between the early and late groups?
It was. Both groups lost a tiny bit of weight over the 2 weeks, which isn't surprising with this level of dietary attention. But the weight loss was more noticeable in the early eating group, about 1 kilogram, compared to less than half a kilogram in the late eating group.
And this difference in weight loss correlated with a calorie difference.
It did. The researchers calculated that the early eating group had a small, basically unavoidable average daily calorie deficit of about 167 kilocalories per day.
Wait, only 167 calories? That sounds tiny. Like, what is that, 2 boiled eggs?
Yeah.
A small handful of almonds? How could that small difference explain the benefits seen in other studies?
It sounds tiny, but it highlights just how sensitive metabolic health, especially weight, is to sustained energy balance. Even that small deficit day after day for 2 weeks adds up. More importantly, it points to the behavioral effect.
Aiming.
Meaning that eating early, finishing by 4 p.m., just naturally makes it slightly easier to achieve that small deficit. It automatically cuts out evening snacking opportunities. So even when trying not to cut calories, the structure itself led to a slightly bigger reduction in the early group.
So the thinking is, in less controlled studies, this effect would be much larger. People eating early just naturally cut more calories, leading to more weight loss, and that's what drove the metabolic improvements seen before.
That's exactly the interpretation offered by the researchers. Professor Ramage put it quite directly, suggesting the benefits in earlier TRE studies were likely due to unintended calorie reduction rather than the shortened eating period itself.
The timing acts as a helpful constraint. The constraint helps reduce calories, and the calorie reduction improves metabolism. The timing itself isn't doing the metabolic heavy lifting.
That seems to be the conclusion from this very carefully controlled trial.
All right, so the metabolic findings were, let's say, underwhelming for TRE enthusiasts. But the study wasn't all null results. Section 3, the circadian impact. Here, the timing did matter, right?
Absolutely. This is the other major finding, and it's a positive one for understanding how meal timing affects our biology. While metabolism didn't significantly change, the body's internal timekeeping system responded very clearly to the shift in eating schedules.
This gets into our circadian rhythms. We know light is the main cue for our master clock in the brain, but other things can influence it too, these zeitgebers or time givers.
Right. And research, mostly in animals until now, has strongly suggested that when we eat is a very powerful zeitgeber, especially for the clocks in our organs, liver, pancreas, muscles.
These peripheral clocks need to be synchronized with the master clock and the outside world. Food timing seems to be a key signal for them.
And ChronoFast provided some really strong human evidence for this. Using that objective body time assay, we talked about the molecular measurement of the internal clock phase from blood cells.
So what happened when they compared the internal clock timing after the early eating schedule versus the late eating schedule?
The difference was quite striking. The participants' internal circadian phase was significantly delayed. It shifted later by an average of 40 minutes after the 2 weeks of late eating compared to after the 2 weeks of early eating.
40 minutes. Just by shifting meals from an 8 a.m., 4 p.m. window to a 1 p.m., 9 p.m. window, you can actually push your body's internal clock almost an hour later.
That's what the molecular evidence showed. It's a clear demonstration that meal timing acts as a potent signal to our internal biological timekeeping systems.
And this wasn't just an abstract molecular finding. It showed up in their sleep patterns, too.
Yes, it correlated nicely. The participants reported through their sleep diaries that their sleep onset, when they fell asleep, sleep offset, when they woke up, and their sleep midpoint all occurred later during the late eating intervention compared to the early one.
So eating later seemed to push their entire sleep-wake cycle later as well. It makes sense if the body clock shifts later, sleep drive would likely shift later too.
Exactly. It reinforces this idea that food timing is a powerful cue for our biological rhythms, as the researchers put it. This has potential implications for things like shift work or jet lag management, maybe.
Did they look deeper at the actual clock genes themselves?
Yeah.
You mentioned the assay measured gene activity.
They did. They saw differences in the expression levels of core clock genes in the blood cells genes with names like PER1 and NR1D1, which are known cogs in the machinery of the 24-hour clock and are linked to metabolism. The pattern of their activity changed depending on whether the participants were on the early or late eating schedule.
So the timing of the food signal was directly influencing the molecular clockwork in the cells.
That's the picture, yes. It provides that mechanistic link.
Okay, so the internal clock definitely responds to meal timing. Did the study find any effects on how people felt in terms of hunger or appetite?
Yes, there were subjective differences too. Specifically, during the late eating intervention, participants reported feeling less hungry and having a lower desire to eat in the morning compared to when they were on the early eating schedule.
That seems intuitive. If your body gets used to not eating until 1 p.m., it probably dials down the hunger signals in the morning.
And they found a potential hormonal reason for this. They measured peptide YY, or PYY, which is a hormone released after eating that promotes feelings of fullness. It's a satiety hormone.
And how did PY levels change?
In the mornings, PYY levels were higher during the late eating schedule compared to the early eating schedule.
So when they were fasting longer into the day, late eating, their body released more satiety hormone in the morning, potentially helping them manage that longer fast.
That's a very plausible interpretation. It suggests a hormonal adaptation linking the expected mealtime to appetite regulation. Interestingly, ghrelin, often called the main hunger hormone, didn't show significant changes between the conditions.
So the adjustment seemed more related to managing fullness or satiety signals rather than the primary drive to eat.
That's what this particular data suggests.
Okay, so summing up this section, time-restricted eating, when calories are kept constant, doesn't seem to be a magic bullet for metabolic markers like insulin sensitivity in the short term. But it is clearly a powerful tool for shifting your internal body clock and influencing related factors like sleep timing and hunger patterns.
Precisely. The chronobiological effects are real and measurable, even if the direct, immediate metabolic benefits of timing alone seem minimal based on this study. And those clock-shifting effects could still have longer-term health implications, perhaps through better sleep or organ synchronization, but that wasn't measured here.
Right, which leads us nicely into Section 4. Implications, caveats, and the path forward. Let's really think about what this study means, but also acknowledge its limitations.
First, let's just hammer home the main implication again.
Yeah.
This study strongly suggests that the benefits people experience with TRE are primarily driven by the reduction in calories that often happens naturally when you shorten your eating window. Energy balance is key.
It helps explain why those early eating schedules might have looked better in less controlled studies, doesn't it?
Exactly. It wasn't necessarily that 8 a.m. to 4 p.m. is metabolically magical compared to 1 p.m. to 9 p.m. It's likely just that finishing eating by 4 p.m. is a more effective strategy for many people to cut out those extra evening calories, leading to a slightly bigger, perhaps unintended, calorie deficit.
So the power of TRE seems to lie more in its role as a behavioral framework, a discipline tool that helps manage overall intake.
That appears to be the most straightforward interpretation of these findings.
Okay, but like any study, ChronoFast isn't the final word. We have to talk about the limitations, the caveats the researchers themselves pointed out. What are the main ones?
Well, a big one is the duration. Each eating pattern was only followed for 2 weeks.
That doesn't seem very long to see major metabolic shifts.
It might not be. While the researchers argued some glucose changes can happen fast, metabolic adaptation, especially in response to a shifted internal clock, might take longer. Maybe the liver and pancreas need more than 2 weeks to fully adjust their function to the new meal timing schedule.
So it's possible that if they ran the study for, say, 3 or 6 months, they might have started to see metabolic benefits from the timing itself, even keeping calories the same.
It's possible. We can't rule it out based on a 2-week study. Longer-term isocaloric TRE trials are definitely needed to see if adaptation occurs over time. The metabolic system is designed to maintain stability. It doesn't change overnight.
Okay, duration is one limitation.
Yeah.
What else? We mentioned the specific group studied earlier.
Right, the study population. It was exclusively women with overweight or obesity, mostly postmenopausal. That limits how broadly we can generalize the findings. Would we see the same results in men? In younger adults? In people already at a healthy weight? We don't know from this study. Their metabolic flexibility might be different.
So we need similar controlled studies in diverse populations.
Definitely. And then there's a really tricky methodological issue, arguably the most important one for interpreting the main result on insulin sensitivity. It's about the oral glucose tolerance test timing.
Ah, yes, the OGTT CanFET. Explain that again. It's complex but crucial.
Okay, so the OGTT involves drinking a sugary drink and seeing how your blood sugar and insulin respond. To get reliable results, you have to do the test at the same time of day for everyone. Because our glucose tolerance naturally varies, we handle sugar better in the morning. So they did all the OGTTs at 9:30 a.m.
Standard procedure makes sense. But what was the problem with that in this study design?
The problem was the fasting duration before that 9:30 a.m. test was very different for the two groups. The early eaters finished eating at 4 p.m. the day before. The late eaters finished at 9 p.m.
So the early group had been fasting for about 5 hours longer when they walked in for the test at 9:30 a.m.
Exactly. And here's the kicker. Fasting for significantly longer can actually paradoxically, make your glucose tolerance look worse on an OGTT.
How does that work?
When you fast longer, your body dials back insulin secretion a bit. It's conserving resources. So when you suddenly hit it with a big load of sugar from the test drink, the initial insulin response might be slightly blunted, leading to a higher temporary spike in blood sugar. It's a physiological adaptation to the longer fast, not necessarily a sign of underlying poorer metabolic health.
Wow. So the early eating group, because they fasted longer before the test, might have artificially looked like their glucose tolerance was worse, or at least not better, purely because of the test protocol itself.
Precisely. This confounding factor could have masked any potential small benefit that early eating might have actually conferred on insulin sensitivity.
That's a huge caveat. It means we have to be really cautious about concluding definitively that early eating has no metabolic advantage over late eating, even isocalorically, based on this OGTT data.
It's a major challenge in TRE research. You either control for time of day and introduce a fasting duration bias, or you control for fasting duration and introduce a time of day bias, testing one group in the morning, one later. There's no perfect solution with the OGTT.
The researchers acknowledge this, right?
Yes, they were very clear that the OGTT-derived insulin sensitivity results needed to be interpreted with caution due to this unavoidable methodological issue.
Okay, one more limitation quickly, sleep assessment.
Right. Sleep timing was based on self-reported diaries. While useful, it's not as objective as, say, polysomnography in a sleep lab. So the sleep timing shifts are suggestive, but rely on participant reporting.
Understood. So given these findings and limitations, what's the path forward? What kind of research is needed next?
Well, the most obvious need is to directly test that synergy question. We know calorie reduction works. Does adding a specific TRE schedule, early versus late, to a deliberate calorie reduction plan provide benefits beyond the calorie reduction alone?
So compare, say, a 500-calorie deficit eaten 8 a.m., 4 p.m. versus the same deficit eaten 1 p.m., 9 p.m. Does the timing amplify the benefits of the deficit?
Exactly. That's a key practical question. And secondly, we desperately need research looking at individual differences. How does a person's natural chronotype, whether you're a morning lark or a night owl, interact with these schedules?
Yeah, forcing a night owl onto an 8 a.m. to 4 p.m. schedule might be stressful and counterproductive, right? Maybe increasing cortisol.
It's plausible. Personalizing TRE based on chronotype, genetics, lifestyle. That's likely where the future lies, rather than a one-size-fits-all timing recommendation.
Okay, let's try to wrap this up and synthesize the key takeaways for you, our listener. If you're trying to cut through the noise on time-restricted eating, what does this landmark ChronoFast study really tell us?
I think the main message is about separating the effects. The study clearly shows TRE is a potent tool for shifting your internal body clock. That's a robust finding meal timing influences your circadian biology.
What?
But if your main goal is improving metabolic health markers, things like insulin sensitivity, cholesterol, blood pressure, or achieving significant body composition changes, this study strongly suggests that timing alone isn't the answer, at least not in the short term. You must also manage your overall energy balance, your calories.
So the popular idea that you could just eat whatever you want, just squeeze it into 8 hours and magically fix your metabolism.
Yeah.
That seems largely debunked by this controlled research, unless that schedule inherently helps you eat less overall.
Right. The mechanism seems primarily behavioral and energetic, not some mystical metabolic switch flipped purely by the clock. Professor Remix, quote, sums it up perfectly. Those who want to lose weight or improve their metabolism should pay attention not only to the clock, but also to their energy balance.
The clock might be a useful tool, a guardrail, but the calorie deficit or energy balance is what actually gets you to the metabolic destination.
That's a great way to put it.
So if you're thinking about TRE, perhaps the best way to view it, based on this evidence, is as a really effective behavioral strategy. It simplifies things, it provides structure, it reduces the opportunity to eat, especially during times like late evening when we might be prone to mindless snacking or high-calorie treats.
Exactly. It shifts the focus from hoping for a biochemical shortcut to using scheduling as a practical tool for achieving self-control and ultimately calorie management, which, as the study shows, appears to be the essential ingredient for metabolic improvements linked to TRE.
Which brings us to our final provocative thought for you to consider. If the main metabolic benefit of TRE really boils down to helping people achieve a calorie deficit by limiting the eating window, does this mean the most powerful aspect of intermittent fasting is simply the structure and restriction it imposes? And if so, maybe the focus shouldn't be so much on finding the perfect window timing, early versus late, but rather on finding the timing structure that you can best stick to, the one that best fits your life and helps you consistently achieve that negative energy balance.
It frames TRE less as a metabolic hack and more as a potentially very effective adherence tool. Maybe less glamorous, but perhaps much more practical and sustainable for many people.
A fascinating deep dive into some really careful science that challenges popular assumptions. Thanks for joining us to unpack the ChronoFast trial.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.